It's a great honor to introduce the first session here. The first paper is by Dr. Yuji Hirao, which is titled Complete In Vitro Generation of Fertile Oocytes from Mouse Primordial Germ Cells.
Okay. I'd like to talk about the oogenesis in the mouse. And oogenesis in the mouse is a lengthy process. Even in the mouse, it takes longer than 1 month, starting from the PG sheet and getting the stage of metaphase 2. And to do that in vitro, it takes the same length of the period of time. And so far, the most successful recapitulation of in vitro oogenesis was from the neonatal oocytes and using the two steps culture systems. And so there is a gap here. So there are breaks in the in vitro oogenesis from the PGC to the metaphase 2. And so this gap, gap have been bypassed through in vivo conditions using the grafting the PGCs to the renal capsule or the ovarian bursa, or by using the ooplasm from in vivo grown oocytes. And so there is a gap to overcome. And these papers suggest that there was abnormal follicular formation and insufficient oocyte growth. And we also lose a lot of oocytes in the culture system from primordial follicles. So if we simply combine these two conditions in tandem, the situation is going to be like this. So the tens of thousands of trials would be necessary and to the success. And the success wouldn't make the trials by others easier, even if it was successful the first time, but it's not going to be easy. So we aim to develop a culture system in which a lot of secondary follicles are produced, and those follicles cannot be just many. Those follicles must be good and healthy at the same time. And it is followed by the follicular culture that sustain oocytes as many as possible until they become competent to undergo embryogenesis. Okay. So I'm talking about the first half of the organ culture. It is organ culture, and the culture period is the culture period is 17 days, corresponding to the 10-day-old mouse. And there are two major events. And regarding the meiosis, the onset of meiosis, actually, they just simply do so in culture. There was not much we did do something about it. And so it is more like asking gonadal somatic cells to make it occur. Sorry, again. And however, the formation of primordial follicle was we found much more complex and complicated. We had trouble. It was a kind of conflict between the good for culture and bad for culture in using fetal bovine coelom in the culture medium. Okay, the organ culture, the concept was very simple. It's placing the placing the fetal goners onto the culture insert membranes and keep them for 17 days. And under the regular culture conditions, it's nothing special. Alpha MUM plus FBS and on the insert membranes in regular conditions. And we thought it would work well, and our plan was to dissect ovaries into individual secondary follicles. But it turned out the use of serum was kind of causing trouble. So this is actually one ovary growing in vitro in the culture system with the medium supplemented with FBS. And ovaries can grow. However, when we take a closer look at it, the follicles are showing some abnormal aspects. For example, they have unclear basement membranes, and so these are the multi-oocyte follicles, and the follicles themselves are smaller than what they should be. So accordingly, isolation of intact follicles at the end of the first culture period was very difficult. So we had to do something about those abnormal follicles. So these ovaries that had been both cultured, and so it seemed that problems disappeared. Actually, no. This ovary was cultured from a newborn, derived from newborn mouse ovaries. So we hypothesized that the problem takes root in the early phase of the assembly of primordial follicles. So to explore the cause of the abnormal follicles, we run RNA sequence analysis, and the sample were obtained from the ovaries cultured from 12.5 embryos after 7 days of culture and newborn ovaries. And the overall general gene expression profile was very correlated. However, we found 547 genes differentially expressed. Geoanalysis suggests the involvement of the AMH and INHA related to the responsible for the primordial follicle formation. And so ingenuity pathway analysis showed some more interesting results. The top 3 predicted upstream regulators were beta estradiol and SP1 and beta catenin. So all those bind to the estrogen receptors and regulate gene expression. So we thought these gene expression are not different because they are cultured for a longer period. It's more like it's mimicking the condition in vivo, the high maternal estrogen conditions. So group studies from Dr. Pepling had revealed that a reduced concentration of estrogen and or progesterone is essential to promote primordial follicle formation. So if this is the case of our in vitro culture, then the experiments we had to do was minimize the potential effects of FES-derived estrogens. So we compared how many oocytes could be isolated from the one ovary at day 17. And the FBS was the primary candidate of the source of FBS. I'm sorry, the candidate for the source of estrogen. And this assumption was supported by the use of SPS, is serum protein supplemented, and with the use of SPS in the place of FBS, the number of follicles isolated from one ovary increased. Then we tried the use of ICI. It's an estrogen receptor antagonist, and it's a downregulator, and which causes the ER to degrade. And surprisingly, the use of 10 micromolar of ICI resulted in more than sevenfold more follicle isolation efficiency from the one ovary. And if we have 7 times more follicles, it definitely changes the our design and scale of experiments for the latter half of the culture period. And also, an decisive result came from the experiment addition of the estradiol together with ICI. Now efficiency was down completely back to the minimum level. Okay, so these were the follicles isolated from the best conditions and the worst condition. And these selected conditions derived from extensive experiments of Dr. Obata at Tokyo Agricultural University. Okay. So these are ovaries cultured under the best conditions and the worst conditions. The improvement of the structure of ovaries, the follicles and everything is better, was evident after the addition of 10 micromolar ICI. When we take a close look, the formation of follicles normal, and actually it became much easier to isolate secondary follicles from these ovaries, and only 3 or 4 could be isolated from this type of ovary. Okay, so it's not only the number that was improved by the addition of ICI. This is a ratio of the ratio of oocyte diameter and follicle diameters. So when ovaries were cultured with 1 micromolar ICIs together with FVS, the follicles could be isolated, but the oocytes became the normal size. However, the follicles diameter are smaller than for the oocytes as compared to the follicles obtained from in vivo grown oocytes. So the follicular growth was retarded in the condition of 1 micromolar ICI. And interestingly, we try to obtain follicles from the medium only supplemented with FVS. It is impossible to collect the sufficient number of follicles to make this graph. So it was, I'm sorry, I couldn't show it. So it's starting from 1 micromolar ICI. Then this concentration was raised to 10 micromolar ICI. Now the ratio of the oocytes and follicles were within comparable within the range of the in vivo grown follicles. So, and this is the final part of the first half of the culture system, and we again run the analysis of RNA sequence analysis, and essentially there are similar results were obtained when we didn't use ICI. But and overall gene expression was similar between the in vivo grown ovaries and in vitro grown ovaries. And in this experiment, the whole ovaries were used, and still 421 genes are different. Oops, sorry. Still 421 genes are differentially expressed, but the point is that now estradiol and SP1 are not in the top of the list of the predicted regulator. So it's now we could eliminate the factors that causing the abnormality of the follicular formation by using the ICI. So now I can start talks about the latter half of the experiment, because now we are ready to collect sufficient number of secondary follicles, and those follicles are not just enough in number; they are also healthy and robust. And as I show you from now on, okay. So the latter half of the culture period is from day 17 to day 31. The basic culture condition is very simple. We use alpha MUM plus 5% FVS and a small amount of FSH, and using the regular conditions for the temperature and air. But in refining the condition for the follicle culture, I think the following 3 points are very crucial. The first was the type of follicle culture. And the second is the use of collagenous treatment. And the third is the use of high macromolecules, a high percent of macromolecules. I use 2% of PVP in this system. So this slide shows the typical morphologies of follicles growing in vitro, and there are various types of follicles, but these are essentially 2 types. One is the oocyte and granular cells exposed to culture medium, and the other is the follicle structure was sustained, and including the basement membranes. So we should call this is a follicle cultures, but these are sort of oocyte granular cell complex cultures. And we use this type because there are a lot of sophisticated follicle culture techniques and development. The development of those follicles have been very good in literature, but we found it very difficult and more demanding to keep them intact for a long period while avoiding atresia, and then to culture them in open structure. So we use the substrate adhering system. So this is the first point. And the second point is a collagenous treatment. And this point is related to the first one, because without collagenous treatment, many follicles remain intact, and it makes the culture system complex. So with the collagenous treatment, in this case 0.1% collagenous for at least 15 minutes, and together with pipetting, follicle structure became disrupted. And we believe that at first we thought we removed the thicker cell layers, but it turned out it remained, as I will discuss later. And so the treatment was done 3 days after the isolation of follicles, and without collagenous treatment, many oocytes survived, but only a small proportion of them developed the metaphase 2, and none of the fertilized. I think that's unavoidable, because our culture system is not suitable for the intact follicles. So this is the second point. And the third point is the supplementation of polyvinyl pyrrolidone. And it should be 360, because if we use 4,000 or something, the size of 4,000 of the osmolarity goes like sky high. So we use 360, and when the PVP was supplemented, oocyte granular cell complex, oocyte granular cell complex were maintained better than in the control medium, and accordingly, at the end of the culture period, a significantly larger proportion of oocytes were recovered alive when compared to the oocytes from the medium without PVP supplementation. So this is the oocyte granular cell complexes growing the condition I have discussed. So we can see the proliferation of granulosa cells and clearly outlined oocyte, but eventually it hides behind the granulosa cells. Okay, so this is what I should note here. There was a continuous production of progesterone, estradiol, suggesting the persistent steroidogenic activity in the culture, the cells in culture. And so I thought we have removed all these thicker cells and basement membranes, and we did disrupt basement membranes, and which was confirmed by the immunostaining of laminin, but the probably thicker cells remained in the culture. So that's why we have a lot of these progesterone and estradiol, but it was delayed until the 27 days or something, so probably we have removed many of the thicker cells, but there are those cells. So we examine the presence of cells expressing HSD3-bin, which is responsible for which is essential for the progesterone production, and without collagenous treatment, those cells are expressing genes localized outer layers of the follicles, and after the treatment of the collagenous, the localization became obscure. But after growth in vitro, those cells are not around the oocytes, I mean the cumulus cells, and other genes that are responsible for the estrogenic activity are also confirmed. So now, finally, I talk about the capacity of oocytes derived from in vitro growth. And so more than 90% of oocytes survived the duration of the culture period and went the meiotic maturation, becoming metaphase 2, and about half of them fertilized normally, and 90% of them developed to the two-cell stage. And the two-cell stage embryos were transferred to the foster mother, and the frequency of the developing pups was ranged between 14% and 40%, and the maximum was 7 pups from one single gonad 1 month before. Okay, so this is good evidence suggesting the recapitulation of the oocyte growth or oogenesis in vitro. Okay. So the sex ratio of the pups was essentially 50-50, and I didn't observe any early demise of those pups. Okay. So then I have thought of some applications, what we can do using this system, and the mass production of oocytes probably is kind of a way, one application. And so it is very difficult to see, but there are a lot of oocytes in here. And so when they are induced to mature, almost all the entire tissues became expanded. And so would you run? Okay, this is emulsified tissues, and so it's 17 hours after the initiation of maturation. So then we use hyaluronidase or something, and we can collect the oocytes with the polar body, the first polar body. And another kind of application is, I have talked about PGC, but probably the same type of culture system could be used for PGC-LC, and from the ES cells or iPS cells. So a team led by Dr. Hayashi at Kyushu University has been working on this project. And so this is summary. So now the oogenesis from PGC to mature oocytes in vitro is possible, and the chain of culture system has been connected. Now the manipulation of observation throughout the course of oogenesis is possible, and we can pick up any stages of oocyte growth, and we can give them any treatment, and we don't have to return them back into the host animals. So we can see it. We can keep watching it, watching them grow in culture dish. So probably it's a great advantage doing the experiments, oocyte growth in vitro. Okay. Finally, I'd like to show my collaborators. And Dr. Morohaku is the first author of our recent paper. And thank you very much for your attention.
So,